Hi, everyone. My name is Nancy Leonard with IDEXX Water, and I'm excited today to talk to you about a recent study sponsored by the Water Research Foundation on wastewater surveillance for COVID-19. We'll be talking a little later with one of the primary researchers from IDEXX that was involved in the study. Before we dig into the study, though, I would like to give a little bit of background about wastewater surveillance and the purpose of the study. Many of you may already be familiar with the concept of wastewater surveillance since it has been in the news a lot lately. But in case you're not, basically the concept is pretty simple. People shed viruses in feces and eventually it ends up at the wastewater treatment plant. Wastewater testing has long been a successful way to detect many diseases, including polio. So it makes sense that it's become a powerful tool for capturing potential community outbreaks of COVID-19. SARS-CoV-2 can be shed in the feces of people who are both symptomatic and asymptomatic, and wastewater testing can detect potential outbreaks up to days in advance. Wastewater testing of raw sewage can serve as an early warning system, if you will, for the community to put mitigation measures in place to help control potential outbreaks. So now that we understand what a powerful epidemiological tool wastewater surveillance can be, I'd like to talk about a study that was conducted by the Water Research Foundation. The study, which is called Reproducibility and Sensitivity of 36 Methods to Quantify the SARS-CoV-2 Genetic Signal in Raw Wastewater, Findings from an Interlaboratory Methods Evaluation in the U.S. I'm just going to call that the WRF study, 5089 for short. But the purpose of the study was to identify methods that produce reliable quantitative measurements of SARS-CoV-2 in untreated wastewater. This was evaluated by measuring the reproducibility and limited detection of 36 different methods being used by academic, commercial, government, and manufacturer laboratories. IDEXX was honored to be able to participate in the study, and I'm really excited today to be able to speak to IDEXX's primary researcher on the WRF study, Dr. Brian Swalla. A little bit about Dr. Swalla before we introduce him, or before we show him. He received his PhD in microbiology from the University of Illinois, where he studied bacteriophage genetics and bioinformatics. Dr. Swalla has over 17 years of experience developing and applying new molecular technologies in the fields of microbial and protein biocatalysts, cellulosic biofuels, and water quality. Brian has worked for IDEXX Laboratories since 2011, where he's a staff scientist focusing on developing new products for water microbiology. He's been involved in developing several validated methods, and he really understands what's required to provide validated, standardized results at scale. Most recently, he led the technical development of validation of an RT-qPCR test for SARS-CoV-2 in wastewater. Welcome, Brian.
Hi, thank you for having me, Nancy.
Thanks for being with us today. So I just kind of gave an overview of wastewater surveillance and the long title of that study. But can you start by just giving us a summary of the WRF study?
Sure. The primary finding was that a variety of methods work for quantifying SARS-CoV-2 in wastewater. Without going too far into the weeds, the study found that there was a high degree of reproducibility among the methods, with 80% of them falling within what would be considered an acceptable band of variability. Importantly, they found that when evaluating wastewater from a single site, lots of different methods work well for quantifying SARS-CoV-2. Another important finding was that many of the factors or protocol differences that were hypothesized to be major sources of variability were actually found to have relatively small effects and were not the primary sources of variation. This included differences such as whether solids were removed from the sample prior to processing, whether the sample was pasteurized before processing, which genetic target or primer and probe set was utilized. At the various highest level, the key finding was that a variety of different methods can produce reliable results for this new kind of testing.
Oh, that sounds like good news.
Yes, I think you're right. Obviously, time is of the essence with this type of testing. So I see this study essentially giving a go-ahead for these methods to be used to start producing data that can be used to inform public health decisions. The last word of the study is the findings support the use of wastewater surveillance for tracking trends in the concentration of SARS-CoV-2 within communities.
All right, great, thank you. Now the study took a look at 36 different methods. But specifically, of course, I'm interested in the IDEXX method. So can you just kind of go over the results specific to the IDEXX method?
Yes, absolutely. I think one interesting finding of the study was that while most methods were able to detect SARS-CoV-2, many of them had at least one non-detect, meaning they did not detect any SARS-CoV-2 in at least one of the 10 replicates that were tested by each laboratory. You can see that in this chart that is based on the data from the study. Each bar represents a method within which the red portion represents the percentage of replicates that were classified as non-detects. This is one way of looking at the repeatability of the methods. This wasn't discussed too much in the study itself, but avoiding non-detects is very important for this type of testing. If you're trying to identify trends, having a non-detect can make your trends more difficult to interpret. Consistency of detection, which includes avoiding non-detects, is therefore very important. As you can see from the results of the IDEXX method highlighted on the slide here in that red rectangle, our method detected SARS-CoV-2 in all of the replicates tested, demonstrating a high consistency of detection. In fact, because we looked at both pasteurized and unpasteurized samples as part of the study design, the IDEXX method actually detected virus in all 20 of 20 replicates that were tested.
Wow, that's pretty good. Thank you. So without having to read the entire study, although that will be available, can you just kind of give me just a couple of key takeaways from the study?
Sure. I think the study itself does a very good job outlining the implications. To paraphrase, if the major finding is that many different methods can be used for wastewater surveillance, then the implication is that labs and customers can start to focus on other factors that are important to them when deciding which methods to use. Just as a couple examples, the study suggested that labs could look for methods with fewer or simpler steps, or methods that require less specialized knowledge to be performed reliably. And that's also the IDEXX perspective. qPCR is always going to be a specialized technique that requires a certain level of expertise. But what we've been focused on from the beginning and since the study came out is making this type of testing as simple as possible by removing unnecessary sources of variation. We're working to eliminate or streamline steps in the process, especially around reagent management. And we're outlining procedures and protocols that are as easy to follow as possible.
That's great. Thank you so much, Dr. Swalla. I know you're a very busy man, so we appreciate your time. We do encourage you to read the entire study, or if you'd like some more information about the IDEXX SARS-CoV-2 wastewater test, both of these things can be found at idexx.com/water-SARS-CoV-2. I hope that you enjoyed our discussion with Dr. Swalla, and I thank you, Dr. Swalla, for your time today, and also those of you out there who were watching today. Thank you so much. For IDEXX Water, my name is Nancy Leonard, and I hope that you enjoy the rest of your day. Thank you.